Now, see, organism consists two types of chromosomes. Autosomes, also known as somatic chromosomes, which are responsible for formation of the body organs, and another chromosome known as heterochromosome or allosome, also known as sex chromosome. So sometimes the twisted word is there: allosomal recessive disorder, allosomal dominant disorder. This type of equation will be asked like that instead of the sex chromosome using that word. Fine. So you remember this. Okay, another point. Can you know about the hanging and megalom? Tell me about Hanking. Hanking discovered X body from grasshopper. Hanking discovered X body from grasshopper, and McClung gave the name X chromosome to X body. After identification of the X chromosome, Miss Stevens, Bridges, and Goldsmith. Okay, so these all scientists united together and give the sex determination theory. So, can I write the Hanking about it? About him? No? Need to write that? Hanking discovered X body from grasshopper. McClung give the name X chromosome to X body. Okay, and then Miss Stevens, Bridges, and Goldsmith these all scientists gave the theory sex determination theory. In sex determination theory, we had learned with the two types of the sex determination, XX, XY type and XX, XO type. So XX, XY type, this is known as ligaseous or ligase type. Okay, and here two types are there, XX, female XY male method, which is commonly seen in Drosophila and human being, whereas XX male XY female. Can you understand this? It is found in the birds. Another method, XX female, XO male, it is mostly seen in XX female and XO male method, commonly seen in grasshopper, squash bug, એટલે માકડ, do નહીં, and along with that cockroach. So XX female, XO male method, it is commonly seen in grasshopper, squash bug and cockroach. Okay? And you know, twisted question, okay? Sex determination in honeybee, the twisted question is haploid-diploid mechanism of sex determination. બરાબર, એટલે એમાં ખાસ ધ્યાનમાં રાખજો, એવું ન સમજતા કે સર ટેક્સ્ટ બુકમાં નથી તો અમે એ નહીં લખીએ. ઓકે, Barbory technique, it is also known as Lyon's hypothesis. Lyon's hypothesis. Fine? Study or analysis of the genetic characters and aspects like genetic improvement among the humans is known as human genetics. Human genetics is also known as eugenics, means well born. The genetic makeup is normal, okay, and Sir Francis Galton, known as father of eugenics. Now here in the human genetics, pedigree analysis there. In pedigree analysis, you will learn with the different symbols. Just little bit we can revise it. Normal female can be represented as a circle. Male represented as a square, and medical status can be represented by the horizontal line between the circle and square, and their offsprings are formed. It can be represented by drawing this tending line in the middle of the transverse line, okay, and the number of offsprings are represented in the number of sequence of the child and the type of the child in the sequential manner in one generation. Just for example, first is square, second is circle, third is square filled with. Then we say that first child is normal male. Can you imagine what I mean to say? Second child is normal female and third child is affected male. We can say like that. Okay, so few symbols which are not given here. Another point is also there. Diamond shaping that number of offsprings are given there, and sex is not specified. Fine. So, what do you mean by this? That in any generation, number of offsprings are there, but no any particular sex determination. Okay, so it can be represented in the diamond shape, and affected persons can be represented, but sex is unspecified. We can represent it in the filled diamond shape. Fine. Now, next point about the twins. monozygotic twins can be represented by this way, and the dizygotic can be represented in this way. If in circle dot is there, this indicates carrier female with recessive character or disease. Fine. So this is represented in this way. If these symbols are there, this indicates death of individual. This symbol indicates abortion or stillbirth. Sex unspecified. Consanguineous marriage between two individuals of the nearest, we can say that blood relation is there. Okay, this can be represented by this symbol. Now here in practical, one practical is there of this pedigree analysis. Yes. So how we identify the child? So we have 4 questions in front of us: autosomal dominant, autosomal recessive, sex chromosomal dominant and sex chromosomal recessive. See, the chart is given there. If one of our parents, if one of the parents, one is affected, if one of the parents is affected and the disease is transmitted into the next generation in both male as well as female, I can give the clue to you. Fine, how to identify this? If one of the parents is affected, diseased, and then in next generation or in their offsprings, male and female both are affected, this indicates autosomal dominant disorder. Okay, this is known as autosomal dominant disorder. And if parents are normal, their offsprings are affected in both male as well as female. So in this way, or in this case, we think about that male as well as female in any generation, it is affected. Their parents are normal, but they are carrier. Heterozygous condition, and one recessive gene from each parent become dominant in their offspring. Okay, so if parents are normal, so from our side we say that it is carrier. Fine, parents are normal and their offspring in that male as well as female affected, we say that autosomal recessive disorder. Understand? So these two charts are there in your textbook. One chart is there of the autosomal dominant disorder, myotonic dystrophy is there, and autosomal recessive disorder in the sickle cell anemia is given there. Another point, myotonic dystrophy means, myo means muscles, tone means muscle has tone power for the contraction or for the performing the particular function, and dystrophy, muscles are dystrophied. We cannot use the muscles for a prolonged time, and muscles become dystrophied, and this disease is known as myotonic dystrophy. The muscles lost their tone for a definite period of time permanent. Okay, and for this dystrophin protein, you know that maximum genes are present in the dystrophin, 2.4 million. Right? We have 30,000 genes. But dystrophin consists 2.4 million. Maximum genes are present inside it. And this dystrophin causes the dystrophy. Another disease is also the Duchenne's muscular dystrophy. Here muscle tone is not affected there. I think you have done this in the 20th chapter of 11th standard. Disorders of the skeletal system and muscular system. DMD is known like that. And sickle cell anemia, we had already learned with this. In hemoglobin beta chain on the 6th carbon, instead of glutamic acid, valine is there. So if the statement is this, if sickle cell anemic carrier parent is there, 50% chances of the normal hemoglobin and 50% chance of the sickle cell anemia condition. Okay, so if you think about that, the sickle cell anemic carrier male marries with sickle cell anemic carrier female, what is the probability of the sickle cell anemia in their offsprings? Options are 0%, 25%, 50%, 75% and 100%. 5 options you have. Sickle cell anemic carrier male marries with sickle cell anemic carrier female. probability 25% only. One become normal, HPA. Two are carrier, HPS, and one become sickle cell anemic, HPS. Not getting. Carrier male marries with carrier female. Carrier, so it consists HPS, right? Both the side you can take it, fine. So you get the result. Contribution of each dominant allele can be identified by this formula: maximum expression minus minimum expression divided by total number of dominant alleles. So we identify the dominant allele contribution in each population. Okay, the proportion is given there, and you have to identify the contribution of each dominant allele. So this is about the 5th chapter NEET. It is over.